Can you close the door? The door.
And don't forget to bring the backup cable, okay?
Thanks. Okay. Okay, so good afternoon. So today we're going to continue what I was talking with you guys 2 weeks ago. And today I'm going to mainly talk about damage pathways in neurodegeneration. And I will basically go into one neurodegenerative disease, Alzheimer, and I'm going to show you, because it's a very nice example in which we can get to the disease from many different places, and it's very illustrative it's also a very common disease, so it's also even useful the knowledge by itself, not as an example. But it's a good prototype to what could happen in a neurodegeneration in which you don't really have maybe only one cause, but you have many things that go together and give you the phenotype. And we will go. And then I'm going to talk the second part of the talk about animal models of neurodegeneration. So this is how we study neurodegeneration, what type of models we use, because we cannot, which is good, we cannot work in humans, which is a good thing, because if not, it will be a little immoral. Okay? So I'm going to check, and I believe Mona talked a little about mouse models of neurodegeneration. So I'm going to focus in the type of model we use in our lab, which is models using flies, okay? Fruit flies, which is a very popular model nowadays. And if we have time, I'm going to talk about polyQ related diseases, which is another type of neurodegenerative disease, which is has been very well studied. But I'm not sure we are going to have time for this. And again, this class is tailored for you guys. So if you believe you want to stop me in something to have 20 million questions, there is no problem, okay? The same with the content of the slides. Don't get scared. Don't get, if it feels it's too biological, okay? I want you guys to get a few concepts that you can use, because this class is built mainly around guests. So the classes I give and Mona gives, the idea is to give a general framework of the few concepts or the few things that are common to everything that you need to use these tools to understand what the people who come and talk about their research will tell us. Okay, this is just exactly what I told you before. So let's talk a little about Alzheimer. So there is basically, we can divide Alzheimer cases in two types. In one case, you have sporadic. This means that you have a person in which you cannot really link, at least very easily, to the family history. Just seems like it's just showing up randomly between people. So maybe your parents never had Alzheimer and you have Alzheimer, or it seems to be no very strong genetic component on it. That's the definition of sporadic. And then the hereditary forms, in which it's very clear there is a mutation which is passed to some of the people in the family, and you can trace it. You can look and you find these mutations. So usually, most of the cases of, for example, Alzheimer are sporadic, most of them. But you have a small percentage which is hereditary. And indeed, in these cases, when we have a hereditary form of the disease, it's very useful, not to cure, but to understand the disease. Because if we know where is the mutation in these few people who have the disease in the hereditary form, then we can make a mice, or we can make flies, in which you mutated the same protein. Or you can, if you know the function of the protein, you can try to think what type of problem is originating the disease, okay? So this is the first thing I'm going to talk a little, and I think it's a very important concept that you guys have to get from this class today. The second one is determining the cause effect in neurodegeneration. So neurodegeneration is something very bad that happened to the neurons and to the brain in general. So it's when you have a brain which has been neurodegenerating, or a brain which is sick and it's neurodegenerating, it's very difficult. It's so bad, this brain. But you can really not say what is the problem. This is like everything is a problem. So you cannot establish very easily cause-consequence. What is the cause of what? And when you see A, B, C, I think I have a slide after that, but if you have A, B, C, D, E, functions of neurons are not working well, you don't know if you have neurodegeneration because this is not working well, or this is just a consequence of the neurodegeneration itself. So it's very difficult to establish cause-effect in neurodegeneration. So another very important factor is what is important of the environment for neurodegeneration. And we talked that in some cases, smoking, for example, some environmental factors may help against neurodegeneration, and some of them may give you more chances to develop these diseases. So this is the three things the thing are important to point out here. So I just talked a little about sporadic and this. I believe one thing that is very, very difficult is determining cause-effect in the neurodegeneration. So in neurodegeneration, we see many abnormalities. So one is oxidative stress. We see insoluble aggregates. We see mitochondrial dysfunction. We see microtubule assembly problems. We see apoptosis, etc., etc., etc. So what is the cause and what is the consequence of the neurodegenerative process? It's a very important question. And of course, in many cases, if you just play with oxidative stress, you can generate mice that is going to have a disease. If you play with the aggregates, you're also going to have it. But this may be just something that is downstream of the real disease. These are like the symptoms maybe and not the cause, but I just speculated because we really don't know. So what we believe now is that it's a little and a little, but this is almost like to say that we don't know anything, no? So the typical approach is to have access to early cases. If you can follow a person since he's born until it dies, and you can see all over the time, if all these pathways are affected, this will be the ideal situation, okay? But this is, I put very difficult. I think this is impossible because you cannot take a person, open the brain when it's a kid, close it again, and see what is happening later. You only have access to the brain or brain samples when the person is dead. So you really cannot, if the person was dead at the very early stage of the neurodegeneration, then the person didn't know that it was, you have no way to have early cases, access to early cases. So indeed there is very big efforts in order to generate readouts from a person that will be early in the, if we can do some test to a person over age, and we can see how this test goes down, we may get a situation in which we may know after a lot of experience which person is going to develop neurodegeneration and which one no, maybe, or some sort of MRI or some sort of assay that we can see from outside. But this is something that people are trying to actively develop now. But basically the paradox here is that an early case of neurodegeneration that doesn't have any symptom to the outside, so you cannot have access to see pathology in the brain of early cases. Is clear? Yeah? So we can also affect the pathway, like I told you. You have 6 or 7 things. You can't see from there? It's too small?
No, I have I lost my glasses.
Maybe you don't want to come maybe more. It's okay? Okay, so the second thing is that it's something like I told you before. You could affect the pathways one by one. We know from what I told you before, many things happen in neurodegeneration. So if you want to know which one of these is the responsible or the cause, you could start to affect one by one and see if you have a phenotype. Well, this is a fallacy it's a fallacy of affirmation of the consequent. It's just obvious because if you have if let's say you may have an effect by altering the axis of distress, you will have an effect in a neuronal function for sure. But this doesn't mean that this is what started the disease.
But something I don't understand is all these things we see every time.
When you take a brain which has of a person who died from Alzheimer.
We see all of these
Yeah.
symptoms.
Yeah.
Like what's different if it's the cause or not? Like I don't get it.
Well, if you want to understand what happened there, how the disease, what is the mechanism of the disease?
Yeah, no, I know, but the question is, if anyway it's going to be, we're going to have the same symptoms, no matter if it's the cause or not the cause.
Well, but if you can cure the cause, maybe you can go back. What you are saying from the medical point of view, what you are saying is rational. You just need to alleviate the symptoms. You don't care about the origin.
No,
but yeah, which is.
It's not a problem, right?
Yeah, it's a smart point, but imagine if you can abolish what is upstream, because then you can abolish everything that comes downstream. Because the idea here, that the idea is that if we could understand these diseases and how they develop, then we could probably point at the person who will get it, almost for sure, and we could give some drug to this person all over his life maybe, starting on his 30s, on his 40s, so he's not going to develop even the first thing in the cascade. Of course, for the people who now have neurodegeneration or are going to have it in the next 5-10 years, we need to also learn how to cure the symptoms, which is also being done. But your point is very good. And because I'm trying to tell you the objective, I'm trying to talk about understanding how the disease is originated. So that's also that's the bias I have. It's more in research than in medicine, but you're right. So the other thing is that looking for inherited forms of the disease and use animal models. That I think is very good, but again, you need to be very careful with this sort of fallacy of affirmation of consequence. Although, if you really know a point mutation is giving this to every person who has it, then this is the cause. Very, very,
When you do an animal,
yeah.
research on an animal, the brain is the same brain. That's something I just don't know. Like our brain and animal brain.
Yeah, of course, neurons are neurons. This.
Yeah, but it's made from the same.
Like, what do you mean?
Like it's built the same. They both have, like say for Parkinson, they both have substation nigra.
It's such in the mouse. At least in the mouse, you have dopaminergic neurons in the substation nigra that are the ones that are more affected when you put, for example, MPTP or this is the pharmacological model of Alzheimer of Parkinson. But yeah, this is the same. And I'm telling you that if you go to a fruit fly, the fruit fly has dopaminergic neurons, and if you make flies that express one of these proteins that are mutated in people with Alzheimer, you are going to kill only a group of dopaminergic neurons, which are in a region that people believe is homologous to the substantia nigra. So, since we believe we are very, we have very sophisticated functions in our brain, no doubt at all. But happily for the research, our neurons are not so different than the neurons from, and the functions that the basic functions are really highly conserved, even if we go to fruit flies. This is very good because if not, the research will be impossible for this disease. Of course, if you work in consciousness, you cannot use even a monkey, maybe not adequate. So the last thing is looking for genetic associations even on sporadic cases. So I'm going to talk about how geneticists do this. But if you have a lot of people which have developed a sporadic case of Alzheimer or Parkinson, and a lot of people who have not developed, then you can find you can find genes that are associated with this. And the idea that happens is that the Parkinson and Alzheimer, maybe they are genetic. They are just multi-genetic. So you need many combinations of alleles. So it's not a simple Mendelian genetics. It's a little more complicated. But you can figure it out quite well. Okay, let's talk a little about Alzheimer. So I'm going to just first talk a little about the sporadic and hereditary forms of these diseases. In the case of Alzheimer, we call the early onset cases, that means the ones that are heritable, appear between 40 and 55 years old, you see, in Alzheimer. And the ones which are sporadic usually appear after 70, okay, 68, 70 years old. In Parkinson disease is very similar, you see, very similar. And in ALS, it's the only thing that the sporadic cases can appear early in life, okay? In the Huntington disease, it's a pure genetic disease. There is no sporadic cases. And if we have time, we're going to talk about Huntington. So what is intriguing about this sporadic against late onset, hereditary against the late onset or sporadic cases of the disease is that they look exactly the same. They look exactly the same regarding the behavioral phenotype, regarding the specificity of the neuronal loss, regarding the aggregate, if you look under the microscope, how the brains look. So what are the differences? So for this, we are going to see this a little later, but I want to just show you how I think Mona showed you all this stuff. That you have two types of, and I think I mentioned last week, you have two types of ask me. What?
No, I just asked if there is a sporadic cases, right? And the second one, which is?
Hereditary.
Yeah, I asked if it comes from heritage.
Yeah. This is the cases in which you can really explain genetically, because you know which is the mutation. And you can do the trees of the families, and like you see that if the mother or the father had it, then 50% of the kids will have it, you know. This is the cases in which you can trace really the disease. Yes?
How can you determine which diseases are sporadic and which patients are sporadic?
Based on the family history. Just based on the family history. If you go to the hospital and they say you have Alzheimer, then they will check, did your mother, did your father have Alzheimer? If none of the two has Alzheimer, this means it's sporadic, basically. Okay? But in some cases, it could be that the father died before the age that could develop even the hereditary. I don't know. You understand? You just look at the history: the brother, the sister, the kids, the brother, the sister, the parents, the grandparents, the uncles, and you can do a little genetic history of the family to see if it's that's the way it is. This is the.
Can't it be that your father died of Alzheimer, but you still have a sporadic case?
Yeah. Because maybe your father was sporadic. Your father can be sporadic, and
with these tests, you would assume that it was hereditary.
No. Wait, if you get it now, and then your father also had it, it depends if you have 12 brothers and none of them have it, I would believe probably it's sporadic. Also depends the age you get it, because the age, usually the sporadic is later in life. The chance of having Parkinson or Alzheimer is very high if you are more than 80. You know, I think it's 50% after 85, something like this. So if you are old, almost it's a huge chance you're going to have it. So if your father had it, I'm not talking like to you, but if my father had it, so then and he was 90, it's likely he was for sure sporadic because he was 90. Yeah.
People who has a history of Alzheimer in their family, will they be under supervision or
Yeah, I believe this depends. There's nothing that we can do anyway. There is nothing that can be done. This person could eat more antioxidants, and I'm not a physician, so I don't. It's a good question, but there is nothing that these people could take, you know. In the familiar cases.
There's nothing that can be done to avoid it, you say.
Yeah, because the sporadic case, the diet, some things you may be able to work with, but in the other cases you can't. There's nothing to do.
But there are drugs to treat the symptoms.
Yeah, you have drugs to treat the symptoms. Yeah, none of them is very good, you know. I maybe it's bad because I'm being recorded. Maybe there is something that I don't know, but there is drugs to take this. But once you start to decline, there is a lot of stuff in clinical trial all the time. So there is hope that we know about this disease so much more in the last 30, 40 years, but even the last 10 years than before. This is going to be already such a big problem for the developed world, you know, that we are really learning about it. Yeah.
So
But there is nothing that you can do. Yeah.
I read in various books that were written by social workers and psychologists saying that early onset Alzheimer patients and sporadic Alzheimer's patients, their behavior is similar but different. That the extent to which you can hope to kind of engage with them and kind of get them to refocus or kind of work with them, with sporadic patients is significantly higher than with early Alzheimer's patients. Have you heard anything about that?
I didn't, but it makes complete sense, especially because they know who knows, some familiar die from it. They saw them die. They saw maybe many of them die, 2, 3, and they know that there is nothing to do. In a sporadic case, if a person is older also, I don't know, who knows, if you have a family, very strong family history, you may be almost like waiting this to happen. You know what I mean? And you know when it happens, when it starts, but I just talking out, you know. I think you know probably better than me. Your point is good. Yeah.
When the disease is considered to be hereditary, is it considered to be autosomal, or there are many genes involved?
No, usually there is one. Yeah. In this case, it is one. And when it's more, we are going to see the gene association studies. Then you have a few, and in one or two of those, only when you combine a few of these mutations, you may have an effect. So let's go to the pathogenesis. You know there is 2 types of aggregates. One that are called plaques, and are extracellular. And then you have the tangles, which are intracellular. And these are hallmarks of Alzheimer's that you see both in sporadic as well as in hereditary cases. I think we talked last time about the APP and how it's a clip and how these oligomers of the APP peptide can, we talk about this, no? Like when I talk with you guys, no? Is involved in this. And we talked that in the case of the NFTs or tangles, the protein which accumulates in these tangles is not APP, but is tau, which is a cytoskeleton protein. Okay. Again, this picture is for decoration. Okay. So the current hypothesis about the pathogenesis of Alzheimer's disease is the amyloid hypothesis, and I talked about this 2 weeks ago, and I think Mona also talked about this. I know that she talked. So basically, we have here, and I want you to remember this pathway, and this pathway will go from the beginning of the disease, where it starts, to the symptomatology and the end. So let's go. Maybe I will just move here. Let's go over this. So we have a APP here, which is cleaved by beta, alpha, and gamma secretase. And depending how it's cut, you remember one of the forms, the 42, the 42 is toxic, while the 38 is not toxic. So depending on the ratio between the 42 over 38, how much is the one respect to the other, you have more or less toxicity. So when you have the AB, and on one hand, if you have a cerebrovascular events like a stroke, you may have more propensity to have these AB peptides. And AB peptides, there is a system that clears them from the brain. So this is a protective mechanism that will clear them out, and it's supposed to be working all the time. There is another mechanism that will lead to the degradation of these peptides. Now, when you have a lot of these peptides, the first thing that is going to happen is that they are going to oligomerize. Oligomerize is that you will have a few of these molecules together, and this is the toxic compound. So this is going to also bind some metals like copper, zinc, and iron, and is going to impair long-term potentiation. So it's going to impair neuronal function, and this is what the A-beta peptide does to the neuronal communication. Okay, impair LTP, impair the neuronal function. Now, when you have impairment of ATP, there is a process that is called fibrillation, and leads to the formation of plaques. Okay. This also, the oligomers also will somehow stimulate the formation of the tangles. And I tell you, the oligomers are extracellular, but somehow they will lead to the formation of tangles. And I know perfectly how it happens. I just don't want to tell you because it's doesn't have. It's not important for this class. Okay, it's known. The signaling is known, more or less known, and in this case, these NFTs form. So now, if you have these NFTs, these NFTs also are helped if there is signal for inflammation and if there is oxidative stress. Okay. Yeah.
These tangles are intercellular.
Intercellular, yes. So when you have this senile plaque, this is a signal for a macrophage to come, signal for inflammation, and it's a signal for oxidative stress. And this has two different consequences. One that I told you, both things, inflammation and oxidative stress, are going to promote formation of tangles. And the other ones is that this is going to lead to the neuronal death. Okay? And the tangle also contribute to this. So this is more or less the hypothesis how Alzheimer happens. Okay? So now you can imagine all the type of mutation that you could have that will help you to have Alzheimer, or that will help you to not have Alzheimer. For example, any mutation in a protein which is responsible for the clearance from the brain is going to give you more propensity to Alzheimer. Anything that helps for the oligomerization or helps for the impairment of the LTP. And we are going to, I took 10 candidates for genes that may be mutated or very responsible for in case of a sporadic Alzheimer. I will show you how these 10 genes are located somewhere in this scheme. Okay, let's start. Okay, this is just the same that I was telling you. So which genes are genetically associated with Alzheimer? So I think Mona put exactly the same slide. Okay? She did? No. She put it, I think she put exactly the same one. So, and these are the most important mutations in early cases of the disease. Okay? Either you have the APP, the amyloid precursor protein, which is mutated, or either you have the preselin 1 or 2, which are proteins which are in the secretase complex. So then it's either the protein which is cleaved, which is mutated, or the protein which is cleaving it. okay, which is mutated. And the early onset happen in a very small percentage of the sporadic of the cases, only 1%. And these mutations account for 5%. Okay, so basically, when you have a mutation in APP, which is this precursor where the A beta peptide is, or when you have mutation in PCN1 and 2, you have more propensity to Alzheimer. Of course, it's not any mutation, it's a specific mutations which are associated with higher production of the toxic form of the AB peptide. Okay. So just, I think, again, that Mona should talk about this. You have the, this APP, which is cleaved by alpha and gamma secretase to give the AB peptide and this intracellular peptide. And depending how the secretase cut, you are going to have different type of A beta peptides. Gamma secretase can cut in the residue 38, 40, 42. So if it's cutting in 38 or 40, it's fine. But if cut in 42, then that's the form that tends to oligomerize and is toxic for the brain. Okay. So when you have more APP, if you have more APP, this is also a very strong case, a very strong factor for developing Alzheimer's disease. So there is a case in you have tandem duplication of the locus. So you have 2 copies or 3 copies instead of 2. And in these cases, you have a dominant disease. This means even if you have it in one, coming from one of the parents, you're going to get the disease. You have also mutations in the promoters. You know what the promoter of a gene is? It's the region that says...
What activates the gene?
Yeah, it's exactly. It's what activating the gene. It's what is telling the gene how much it needs to produce of a gene. So it's not in the part that is encoding the protein, but it's upstream. So if you have mutations in the promoter, which this make this promoter to be more active, then it's like having 2 copies. You have more amount of APP, you have more amount of the bad A beta peptide. And as I said, everything is about how much A beta 42 you have in relationship with the A beta 40. Okay, this is the same thing. So, okay, let me just say this doesn't matter really. Okay. Indeed, you know that the, I don't know why it's not in the slide, but this the APP is in the same chromosome, which is duplicated in a Down syndrome. Okay, when if you have Down syndrome, you have trisomy of 21, and APP is also in the 21. So if APP really gives more Alzheimer, we will expect people with Down syndrome to have Alzheimer, and the correlation is very, very high. Yeah, so people with Down syndrome, not only they have Down syndrome, but they have also Alzheimer. Okay? So are the sporadic forms of Alzheimer's disease hereditable? And this is seems like a very tricky question, because we are saying that it's sporadic. We are saying it's not hereditable, but doesn't matter. You could have a disease which is sporadic, which means that you cannot predict when it's going to show up, but which is caused from hereditary pieces. So if you have a disease which is caused by 4, 5, 6 mutations that have to happen all together, you're not going to be able to trace it very easily through the family. But if you do the correct calculations, you could do it. So what people try to see, they took twins, a lot of twins, I don't remember how many, is here, 11,000, no? 11,000 twin pairs, okay? And they look whether these people, what is the likelihood if one of them develop a sporadic Alzheimer, or the other one will develop a sporadic Alzheimer, and vice versa. And this show is very, very, very high. It's 79%. So this means that Alzheimer is hereditary. It's not just what you eat or what you don't eat through your life, or where you work in a place, or where you sleep at night or not, or maybe this stuff has some influence on it, but seems like a lot of the disease can be explained by the hereditability, okay? It's very complex hereditability, but it's genetic. So the question is then, which genes are linked to the disease? Which means also to say how the disease is caused. So this I want to go to explain you. You guys know what the association studies are? No? Okay, it's good. It's good for me, because then I can explain to you. Because I think it's a very nice, it's something that geneticists do. It's a very nice approach. So if you have a group of people which are affected by a disease and a group of people which are unaffected by the disease, the association study is going to test whether a particular allele occurs at higher frequency among the affected than the unaffected individuals. So if you have 1 million of persons with the disease and 1 million of persons without the disease, you are going now to look at all the alleles of these 1 million people with the disease and all the alleles of the 1 million people without the disease. And I have no time to explain you how you do that. Yeah, there is a huge database of SNPs that is more or less like all the alleles, more or less, okay? And now there is new technology, you can even do it better. It's not per person, it's not very expensive, but if you want to do 1 million, it's very expensive, but it's doable. People is doing that. So this is what we call a genome-wide association study, okay? And I just wrote the stuff down. So a genome-wide association study is an approach that involves rapidly scanning of markers across the genome. And the idea is that these markers are associated with the different alleles. So for examples, of course, you need a very large number of subjects, because when I talk about SNPs, SMP, we talk about single nucleotide polymorphism. This means a position in the DNA in which people have different nucleotides. That's what we call alleles, different alleles. Okay, sure? Sure? So you really need a lot of people, because in many cases you may have this allele and this just by chance you have or not have the disease. So you need to have a huge amount of people. So basically what you do is like this. Let's say here we have 4K. I don't go through the text, but you guys can go afterward because it's self-explanatory. If you have, let's say, 4 people with the control and 4 people which have Alzheimer, you can see like this, but multiply by, I don't know, 50,000 for all the alleles for all the genes. And you can look, for example, this allele can take the form of 0, 1, or 2, okay? But look like doesn't matter what you get, that you are controlled, and doesn't matter what you get, that you have the disease. So there is no correlation between having this allele and having the disease. Now, look, Andre, if you go carefully over all of them, you will see there is no one in which you have a good correlation, except for this one, in which all the control have 0 and all the case have 2. And from here you can see that why to take 4 cases is better than to take 1 case. And you can imagine now why to take 50 cases is better than to take 4 cases. If you have 50, probably not all the people with the disease will have a 2 here. Some of them will have it, some of them will not have it. If all of them have it, then it's like a mutation that for sure gives you Alzheimer's. So it's not all or nothing in this case. It's more like you could have a set of mutation on this other set of mutation will also give you the disease. You guys understand the approach? So using this type of approach, people have done many of them. This is, look, I want just to show you the different platforms that people use. In this case, you can look at 500,000 different alleles, or 470,000 different alleles. We are not going to go into the detail at all, but I just go tell you the most important or most significant target of all these studies. Every single one, this is way 10, 15 times more significant than everything else, is apolipoprotein E. Okay. This is the protein. Okay. And the idea of the apolipoprotein E, ApoE, has to do with the transport of cholesterol. Okay, that's the function of the protein. And apolipoprotein E, there is some mutations that will make you more prone to have Alzheimer, and some mutations that will protect you really very much not to have Alzheimer. Okay. And I'm not going to go into the functions, but basically there is a clear relationship. Yeah, I have it here. If with the chance, the amount or the allele of apolipoprotein E that you have has a very clear correlation with the possibility of cerebrovascular events that are going to be also linked, as I told you before, with the production of a beta peptide. Also, the clearance of the brain is very of the particles is very depending on apolipoprotein E. 1 second. And the oligomerization is also, yes.
What's cerebrovascular?
Like a stroke. Okay. And this is the most clear one. So by now taking all the cases, I told you many people have done different studies. You know, one in the States, one in Germany, one here in Japan. Well, you know, many people have done in many countries, many different populations. So what a few years ago was done was to integrate all this data. And each time a new study on Alzheimer comes out, they get the data and they integrate it there, and they can rank the top candidates. And we're going to just go over the 10 top candidates, okay? So their names, the 10 top candidates, and I'm going to show you where they are acting and what they do. I think, as I said, this is a very nice example of, and we are trying to do this quick because I see that it's like we are more than halfway. Yeah, you have a question? You have a question, no? Ah, you asked me before, okay. The first one is ACE. So ACE is an angiotensin converting enzyme 1. And I don't know, this is a enzyme which is belongs to the family of enzymes that are called metalloprotease, and that is involved in the regulation of blood pressure. And the mutations on it increase by 20% the chances of having Alzheimer. So where do you think it's going to be? If it works in the blood, something is wrong here. If, yeah?
And in these cases, you are sure about the cause and effect?
Well, no, you know, it's correlation, you know.
So, but here you said that it increases the
Yeah, no, in this case, you know, I'm not a geneticist, so I don't believe it's a cause effect. But it's cause effect. That's the geneticist will tell you that it's cause effect, and it's let's say if you have the gene, you have more chances of having the disease if you have the mutation. But the way you know it is really just because you know that the person with the disease have more this allele. So it's also really, if you think logically, it's a fallacy of affirmation of consequence. But I think you cannot really do anything if you stick so much to the logic. And there is a point that you have to be more, a little more lax. But in this case, because the numbers are so big, this is a, how do you say? In science, it's not a philosophy. You cannot apply it to, you know. But of course, it's because you're taking the data and then you say this is the cause. It's not a. okay. And then there is a very good evidence for ACE being involved in the disease, because if you take a beta peptide and you put it in a tube, and you take ACE and you throw it there, ACE is going to degrade it. So this suggests that maybe ACE is involved in the degradation of the peptide. But really no one did the experiment in vivo, or I don't know, maybe people did, but it didn't work the way that is not published at least. So we do not know about it. And ACE also have a role in blood pressure control. So then ACE is also here, because can influence or can increase the chance of cerebrovascular events will increase a beta peptide production.
So, can I ask?
Yeah.
There is a class of drug called AC inhibitors, which is used to control blood pressure. So taking them can.
Yeah, this is. Yeah, so this is one of the. It's not there. It's here. Inhibitor of ACE have been used with some success, and the mutation should increase. And some success. It was easy to do this test because the drug was already approved. So then it was easy to do the trial. So and it has some effect. So then this second enzyme called cholesterol hydroxylase, is involved in the synthesis of cholesterol. So again, like apolipoprotein E is linked to the cholesterol metabolism. And this we know is associated with higher levels of A beta peptide. And then it's in here, okay? And also the clearance. The production, and because it's cholesterol, the cerebrovascular frequency has to do with these two, okay? I'm going to, now you understand what I was trying to do. I want to tell you the pathways and the mutations are all associated with these pathways we saw at the beginning. So I'm going to go quick now, okay? This is the third one, nicotinic acetylcholine receptor. And people believe that there is a change in alternative splicing of the receptor by the mutation. And this indeed is is here and here, okay? So we know that the impairment of the LTP is mainly in cholinergic neurons in Alzheimer. So then the acetylcholinesterase mutation may have to do with this. May increase the sensitivity of how do you say? The sensitivity that the neuron or the synapse or the channel has for this inhibition. Even with a smaller amount of the plaque, you are going to be able to inhibit. And then also for the final pathway from the tangle to the cell that seems to be involved. Then we have cystatin C, which is an inhibitor of a type of protein called cysteine protease. And this is involved in this part of the pathway. We know that the mutations in cysteine lower the secretion of the protein up to 50%. So it's like a loss of function. You lose the function of the protein. And the idea here is that you have less or more production of the plaque. Okay. As we go down in the list, the probability of this association with Alzheimer is less and less and less, okay? So in this case, change in GAP2 expression could affect the formation of the tangle. This is mediated by a GSK3 kinase, okay? It's here. And it's also regulated in AB formation, so it's also there, okay? The laminin, really laminin, they don't really know, I was putting it, they have no idea how this is not linked to any of the pathways I showed you before. They really, it was found very, very significant in 1 or 2 studies. That's why it's so high in the list. But it's not clear what is real or not, okay? Then tau, and I'm not going to talk about tau, although I put a few slides here. Tau is a cytoskeleton protein which is involved, is very important for the trafficking of vesicles. You know, neurons are very long, okay? Especially the ones who have very long axons. So you need the stuff for travel from the body of the neuron to the terminal. So the trafficking is very, very important. And tau is involved in this trafficking. So when you have aggregation of this tau in the terminal, the stuff cannot travel anymore, okay? And it doesn't matter. I just want to say this. This is a protein called prion protein, which is also involved in another disease, inclusive Charcot disease or the mad cow disease. And it's known when you have mutations in this protein, you also have more propensity to Alzheimer. And this has to do with with the formation of plaques. Okay. This is sortilin, and this one is working in the production of AB peptide. And the last one is transferrin. It's involved in iron metabolism. And I told you that this aggregate have a lot of iron, so this, in this case, is involved. In this case, is involved with the formation of the tangle. Okay. That's okay. What I wanted to show you is what we have a model, and most of the mutations which are associated, strongly or weakly associated with the disease, have to do with this model. Okay, and this is, of course, the things go in two directions. No? We build this model a little based on this or observations like this, and this observation sharpen a little this model. But the idea is that you need a few of these mutations in order to get Alzheimer. Okay. So this, for now, you should have an idea or a concept of what are the type of things that happen to a cell in order to develop Alzheimer. And I hope you have it. And once if we know that this or this or this or this pathway may be involved, we need to test it. And there is many ways to test it. One way is to have the neurons in a dish. We can culture the neurons for a few days in a plate, in a dish, and we can see now if you inhibit the pathway A, B, C, and D, if you have some morphological features of a neurodegeneration. But this is not the best model because you have the brain outside the animal, the neuron outside the brain, and even farther from the animal. So animal models, and you can follow the animals alive, usually are much better. And there is a lot of work done in mouse. And there is, in the last few years, a lot of work done also in the Drosophila, which is, Drosophila is a fruit flies, which is a very useful model in general, but also for studying neurodegeneration. So why people like it so much to use Drosophila? The first thing that they have a very short half-life. So the half-life, you know how long it is from the moment you have an egg until you have a fly? You have any idea? It's 12 days, okay? So if you want to make a transgenic fly, it will take you 2 weeks, 3 weeks to generate a new fly in which you can introduce the transgene you want. For example, the one which encodes for a protein which is involved in some neurodegenerative disease. If you want to do the same in mice, it's going to take you 5 months. And the genetics is very, very complicated. So in order to have the right genetic background, it's going to take you at least a year. But here in a month, you can do it. Even if we don't like it, the neural system of the fly is not so different than our neural system. So most human diseases have a gene in Drosophila which is very, very similar. Okay? And indeed there is there is a database of all the human diseases that have been found in Drosophila. It's most of them. If you know how many genes are in the human genome, how many idea? 26? No, a little more. It's like 30,000, something like this, okay? No, you were very near. So Drosophila, you know how many genes it has? 15,000, 15, 16,000. You won't know how many rice has? 600,000? No, I think it's like 50,000 or 70,000. It's much more. And you know, there is no correlation really between the amount of genes and the complexity of the organism unless we don't know something very special about the rice, which I don't know. Only that it's tasty. And for example, the worm. Worm is not as sophisticated as a fly, but the worm has 20,000 genes instead of 15,000. Still, if you still could tell me, okay, in humans we have twice, in human or mouse we have twice the amount of genes that in flies. Why is this? This is because in humans, usually you have for each single function, you have 2-3 genes that can do the same thing. However, in flies, you usually have only one who does a function. And you know, this is something which we can say, okay, then the fly is not useful. But I believe this is something that makes the fly much more useful. Because if you take a mouse and you have 3-4 genes that can do the same function, and you mutate one, many times you see no phenotype, no effect. Why? Because if you mutate one gene, then another one will replace it. But if you do this in a fly, when you mutate one gene, there is nothing that can replace it. So in many cases, this has been proved to be very good. Also in flies, you guys are falling asleep, you okay? Yes, you're falling asleep? No, you're okay? Okay. So using the Drosophila has another advantage, is that you have amazing genetic tools. So basically you can, using some tricks, this is the brain of a fly, okay? And we can express basically whatever we want. In this case, we express a protein which is green. But we can express whatever you want in everywhere you want in the brain, from a group of neurons that do some function, for example, memory, to a group of neurons that do something else, to even single neurons who do something else. So we have the power we can generate animals that express whatever we want in the place we want and in the time we want. And you cannot do this with other model animals. So it doesn't matter exactly how you do it. Not only you can express whatever you want in the time you want, whatever you want in the time you want, in the place you want, but you also can down-regulate. You can inhibit whatever you want in the time you want, in the place you want, okay? And there is available resources that you can get transgenic flies. You can just order them and get them for like $50 each one for any of the 15,000 genes that exist, so you can down-regulate any single gene that you want to do it. And the same for overexpress, to put more of the gene. Okay. So how can we use Drosophila in order to model neurodegeneration? So there is two types of I would say, in which neurodegenerative disease can be inherited or models can be done. In one case, you have models in which you have dominant neurodegeneration. So if you have a wild-type function, if you have a mutation that gives you a dominant effect over, even if you have wild-type background, okay? It's like in a person, if you have one allele which is bad and one allele which is good, you're going to get a disease. It's a dominant disease. In this case, what you do in the fly is that you just express this protein of the disease, which is dominant, in a wild-type background. Because the idea that these dominant diseases is not that something is missing. It's that the protein which is mutating, what is mutating, is doing something bad per se. It's not just that you are losing the function of this protein, but you have a protein which has a dominant effect. You understand me or no? You don't? Okay. In the other case, you have recessive neurodegeneration. In a recessive neurodegeneration cases, you have mutations which inhibit function of a protein. For example, a protein which is cleaving or degrading the aggregates, if you have a mutation on it, you are going to degrade slower now, okay? But you really need to mutate both alleles in order to have much less degradation, okay? So in these cases, what you do, you usually need that this gene also exists in the fly, and you just inhibit this gene in the fly. Hopefully that this will recapitulate what happened in the humans. Okay. So the question is, let's now think that we have a toxic protein. A protein that when it's mutated, is provoking the disease, but it's provoking the disease because the protein becomes toxic. The protein is dominant. So where do you express this toxic protein, and how you can follow the neurodegeneration? So you know that one of the most intriguing things about the neurodegeneration is that if you have a protein which is toxic, the protein is being expressed in our body, in all the body, but only a few places in the brain are affected. This is just a note. So you could just express in all the neurons, well, and just follow whether the fly is dying earlier, like the people with the neurodegeneration. You could express it only in some neurons. It's horrible, this thing. Okay. You could express it only in some neurons, and then open the brain and look if the neurons are there to see if the neurons have died or the neurons have a morphological problems, etc., etc., etc. But it's a lot of work. And but you can also just express it. What people have been doing is people have been expressing them in the eyes. So the eyes of the fly, you can see them from outside. They are neurons themselves. They are formed by neurons. And then you are going to show you pictures. If you express a neurotoxin like this in the eyes, then the eyes is really in bad shape. The eyes is really losing layers of cells. And so then you could see from outside, from a fly which is alive. And you could follow over time how, why you put this face? I will show you, don't worry. So maybe you shouldn't look. So the fly eyes have a very organized structure, which is very, very sensitive to expression of neurotoxins. And don't look now. If you look, this is the eyes of a fly. Okay. And this is the way this look doesn't matter. Okay. Now you don't have to look, because now I'm putting the picture. And this is but this is not so bad as the one afterwards. So then if you look at the eyes of a wild-type fly, it looks like this. Look how organized. And you have these hairs. Hair, you see that it's coming in each well done. You okay? And then when you, this is also wild-type. It just is expressing a protein which is a little toxic. And you see the eyes look fine, and the hairs are there, okay? Now, if you express this one, which is a polyQ protein, look how the eye is completely, raw, okay? So it's not in good shape at all. And the cells are neurodegenerating. And if you look at now individual cells, you see there is a vascularization here. The cells are in very bad shape. The cells are going to die. And for doing this, you really don't, for looking at the anatomy of the eye from outside, you don't need to kill the fly. You just can look at it. You can put the fly to sleep and look at it, okay? So this type of model has revolutioned the study of neurodegeneration, this type of study in the eyes, okay? So it's over.
Okay.
Yeah, I think I have one picture out, but I will tell you before. So what can we do with such a model? And what we can do is identify genetic modifiers of the neuronal toxicity. So what people do is they generate flies which express this neurotoxin in the eyes. And I told you that in Drosophila you have the power that you can express whatever you want in the place you want, in the time you want, or you can suppress whatever you want in the place you want, the time you want. So imagine if you know nothing about a disease, and you have this fly with this phenotype in the eyes. Now we could try to express every single gene, 1 to 15,000, one by one there. And we can try to see if when we overexpress the gene A, B, or C, if the disease gets worse. This will mean that this gene is in the pathway of the disease and is contributing somehow to the output. But if the disease gets better, probably means that it's also in the pathway of the disease and that the disease maybe is affecting this pathway. Let's say if the disease is inhibiting protein A, and you now put a lot of protein A, you may be able to overcome this inhibition. So in this way, with knowing zero about the disease, only which is the mutation, you can get what are the pathways involved in the disease. And this has been amazing because a lot of what we learn about tau phosphorylation and the formation of tangles, we know it from the experiments done in flies. It's the same with some modifiers of poly-Q toxicity, Fragile X. Many diseases we are learning a lot about this. Okay. That's what I'm trying to tell you. This disease or this type of model have been very, very useful to discover pathways involving neurodegenerative diseases. Don't look now. Okay. So this is a case, for example. You have a fly now which is wild type, and this is the wild type, or this? I think this is the wild type. I don't remember how. I think it happened anyway. Yeah, we don't have the one with the one with the disease, the one I showed you before, which is very, very bad. You remember? Very, very bad. So these people have done a screening and have found other mutations like this one, which make it even worse than before. And other mutations, in this case, are all mutations that make it a little better. You see, this is almost this in this eye also expresses the neurotoxin, but in addition to the neurotoxin, it expresses some other gene, which make it a little better. So, using this type of screening, you can find enhancers of the disease or suppressor of the disease. And this is very important because it reveals new pathways that you didn't know they are related with the disease at all. Okay, this is another example. This is the eyes of a fly, and this is the control. This is other thing that is related with the disease is that the neurodegenerative diseases are progressive. So in the day one, in young people, in people who start to develop the disease, it's not as bad as people who have been having the disease for a long time. They get worse and worse and worse. And it's the same with the fly. This is just an example of the section of the eyes 1 day after being born. And 1 week after being born, you see, this is much worse than this. And what the eyes is doing, the eyes is giving you perception of the light. So one thing that you can do, if the eyes is not working well, the flies usually, when you shed light to them, they move. So if the eyes is not working well, the idea is that the fly should move less in response to light as the disease progresses in the eyes. And that's what I see here. This, in this case, is not the escape from the light. This is the going to the light, but it's a similar idea. If you have a control fly, there is no difference where the flies are young or old to go to the light. But when you make the eyes sick, you can see that they already, when they are very young, they don't go much to the eyes. But even when they are older, and this is only 4 days, you know, very little, they already are not going to the light anymore. But the question is, this is very nice, and I tell you that this is very popular, and there is many success stories with this type of approach. But this is a screening like this, ideal. That's what you really want to do. That's the best we can do or not. So it's because I changed it from the other computer. There is a few advantages to use this system. One, that is very easy, because you just need to look at the fly and you can tell if the fly is in better shape or in worse shape than in the disease condition. Also, the eyes cells are neurons themselves. It's not that you are doing this in other cell type. The eyes are neurons. And in this way, also, you avoid to do dissection plus staining in order to see if the neurons are alive. And you can imagine if you need to screen for 15,000 genes affected, you cannot open the brains to look because it's just not feasible in 20 years, I don't know, of work. The problem is that the eye neurons are a very special type of neuron, which usually is not affected in this type of diseases. And there is some evidence that the eye neurons may be more resistant to other type of neurons to the disease. And they assay cell death, not cell impairment. And we are interested in understanding the progression of the disease, not just the end point of the disease. Yes?
Another thing I wonder is we looked at, I know we looked at the twin study and it showed that Alzheimer's does normally show up in twins. And so it has clearly a strongly genetic basis. But can it be influenced by environmental factors? And if so, can you at all accurately assess the interface between environmental factors?
Yeah, also the twin studies is a problem, because they usually grow up in the same environment.
Exactly.
Yeah, in a very, very same environment. So yeah, there is many studies with the environment, but it doesn't seem to be very, 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 very strong, That's the studies that there's a study showed, if you eat this, you eat that, if you are in this condition, this other condition, you have 1% more chance, 2% more chance. We're talking about very small influence, but maybe there is something we don't really know yet, and that will prove very important. And this type of thing can be done with a system like this one, because there are a lot of screening going on now with drugs to see whether you can, in a normal fly or normal mice, develop neurodegeneration just by eating or being exposed to different compounds over your life. Okay, just almost, have 10 more minutes. So what do we need in order to perform an enhanced suppressor screening for modifiers of neurodegeneration in the brain? So we need to develop a new way to monitor the extent of the neuronal surviving or death from intact animals. And it will be much better if we can have a system that more than telling you that if the neurons are dead, it's telling you if the neurons are working properly, okay? And I tell you that in my lab, we have developed this type of model following this principle. Wow, this is. So what we do, if we restrict the expression of the neurotoxin to very, very few neurons, even the 16 neurons, only 16 neurons in the brain. Now, these 16 neurons in the brain are doing something very, very, very special. Okay, I will tell you what it is. So then, if the function of these neurons get impaired, and if this something very special they are doing, the task they are doing, now they don't do it so well, this means that we can use such a model, and this is exactly what we did. On the side, we have a second way in which we can count, or we can know how many neurons we have alive by some other method that also we can track in vivo without killing the animal. Okay. And these neurons that we talk about, in this case, this is the fly brain, are 16 neurons in the brain, which are here. These are in two sides. And these are the neurons who control circadian rhythms. Do you know what circadian rhythms are?
Yes.
What?
Sleeping and
Circadian rhythms control the sleep-wake cycle. Yeah? You're going to say something? No?
also the seasons of
They are also related with the seasonal adaptation, which is very important for the animals who do hibernation, for example, and to
Also menstrual.
very nothing known about that. No, they very a lot of evidence that may have to, but no one really did it in a very.
But what if it has to do with everything that is cycling?
Yeah, I agree with you, but just the menstrual thing has not been investigated properly, but many indications suggest that it has to do with this. Even with aging, may have to do too. But the idea is that we have a system that is counting time, and this system is independent of the light. I mean, this count 24 hours independent of the light-dark cycle, although the light-dark cycle can influence it and can reset it, okay? When you guys, if you go to Japan, hopefully not these days, or to America, then you experience something very bad for a few days because your body believes that it's in a complete different light-dark cycle, okay? But after a few days, your body gets entrained to it. Usually it's when you have to come back, but hopefully it's not. And then you can, what I mean is that it's independent of the light-dark, but it's entrainable by it. And if we take a fly, it's a fly, and we put them in a chamber that simulates light-dark. So this is 1 day, 12 hours, 12 hours, 12 hours, 12 hours. And you can see the fly moves a lot in the morning, okay? You see? This is plotting the activity, the movement of the fly.
The locomotor.
Yeah, the locomotor activity. So you can see the fly moves a lot in the morning, okay? Then take a siesta in the middle of the day. And then just before the light is going off, before, the flies start to move and move a lot. And the idea here is that the fly can anticipate. You see, the fly knows that soon the light is going to go off and start to move before, okay? And when it's the night, the fly goes to sleep, sleeps, and the day after wakes up again. And if you now take the light off, it's like putting the fly in the bunker or in the sunway, okay, underground. Then you will see the fly keep moving at the same time. This means that it has a way to know when it's the same time, and this is the clock. So what we do basically is, this doesn't matter, we just show you the example. We just express we express a lot of different neurotoxins in these neurons, and the first thing that we saw is that if we express them for a long time, and we express a lot of it, we can kill the neurons. So the neurons usually are in these positions in the brain, but when you express, the neurons are gone, okay? The neurons that were here, this is a very strong neurotoxin and a very long exposure. If you have the neurons supposed to be here, but when you express the neurotoxin, you have no more neurons anymore. And what is more importantly, you remember that I told you that the flies move in the morning and in the late afternoon, and when you put them in constant condition, they move every time at the same time, every day at the same time. Now they look completely, they completely lose the sense of time. The flies are completely healthy. They have no problem. The only thing that they completely lose the sense of time, and we use this system, which I think is not only myself, but we believe it's very good. And yeah, we show that the neurons are there. The neurons are sick rather than dead, because we can see the neurons by using other neuronal markers for these neurons, but the neurons are not performing the task that should be performed, that is to generate circadian rhythm. So, okay, so I think that is that's it more or less. So, you guys have any question? Any question? No? Okay. So I will see you around. I think